Well, hello everybody. Hope you're doing well today, and that you're as excited for this Glow Lab as I am. I've got my green fluorescent shirt on to help get ready for the pre-lab here. So what I'd like to do is sort of walk you around, let you see what is set up at the lab stations. So we're going to have numbered lab stations. So if we go to a particular location, here you'll see the number for the lab station, equipment that you'll use. We have these inoculating loops. You can see here, there's like a little circular end. It's like a little wand for blowing bubbles when you're a little kid or now. But you're going to use these inoculating loops. We're going to have Petri dishes with plates set up for different types of scenarios. So LB stands for Luria broth. Luria broth is like food for E. coli. So we're going to have some plates with agar. I've been pouring the plates here today. So we have plates with agar that has been melted and is now hardening up inside the plates. But the plates are going to be like a medium that is food and home for the bacteria. Okay. So we're going to have different types of agar plates available. Some agar plates are just going to be Luria broth. They're just going to be food for the bacteria. Some plates will have different types of antibiotics, either kanamycin or ampicillin. Another piece of equipment you're going to have available are these transformation tubes. Sometimes they call them culture tubes, but or we're going to use them for bacterial transformation, so we'll call them transformation tubes. And then you're going to have these pipettes. Okay, so that'll be at each lab station. At the front of the room, we're going to have the plasmids. If you'll recall, a plasmid is a little circular ring of DNA, and it's going to have different genes of interest in it. So on ice, I'm going to have some plasmids up here, and each group is going to be designated a particular plasmid. So you'll look at what your group number is at your lab station, and then that'll correspond to a certain plasmid that your group will use, either plasmid 1, 2, or 3. Okay. So your group will use that particular plasmid in your transformation experiment. So again, I'll keep the plasmids up front. I'll also have some starter plates that have bacteria growing on them. At the end of this weekend, I'm going to come in and plate some bacteria. They're in the fridge right now. People don't know that their lunch is in the same refrigerator as some E. coli. But I've got the bacteria in the fridge right now. I'll come in and put them on some starter plates and put them in an incubator so that the bacteria grow. So here's the incubator. I'm just getting it up to temperature here so that it's running at about body temperature because that's, what E. coli like. So we'll get the incubator running at body temp. I'll come in and plate the bacteria, and then you'll grab them off the starter plates and do your bacterial transformation. At the end, you're going to need to spread the bacteria out. And it's sort of fun the way you're going to do that. We've got some little glass beads. So you're going to pour some. At the end of the transformation, you're going to pour the glass beads on the plates and spread them around. Now, I've jumped around a little bit here just to try and talk about equipment. Don't, sweat procedure just yet. We're going to talk about that stuff. So what I would like you to do is, if you would, please grab your lab packet. I'd like to go through part of that now together. I actually have to go get mine. Alrighty. So I strongly encourage you to read through the background information because, again, this is a really time-sensitive lab. Absolutely, this is the most technically challenging lab of the year, and you got to do the right steps in the right order at the right times. Otherwise, you won't get the transformation happen. You don't get glowing bacteria. So I strongly encourage you before lab on either Monday or Tuesday, read through everything so that you're sort of familiar with what's going on. So read the background information about transformation. So that's pages S1, S2, S3. Again, it goes through all the components of the lab. So please make sure you're going through that. So now what I'd like to do is pick up on page S5. It has the pre-lab information. So let's move to page S5 and let's sort of read through what's going on where it says pre-lab inquiry. It says you work on a team in a research lab. You have 3 separate plasmids in your lab, but the labels have come off the tubes and you no longer know which plasmid is which. Come up with an experiment that will give you the definitive answer as to which plasmid is which. For the time being, you'll refer to the plasmids as plasmid 1, plasmid 2, or plasmid 3. What you need to know is that one plasmid has a kanamycin resistance gene. So what that means is if you have an agar plate with kanamycin, so again, we'll have these petri dishes with some agar growing on them. If you have an agar plate with kanamycin and the bacteria grow on that, that means they've got the plasmid for kanamycin resistance. And then two plasmids are going to have an ampicillin resistance gene. So that means they'll be able to grow on plates that have ampicillin. In addition to having an antibiotic resistance marker, one plasmid also codes for the gene for green fluorescent protein, or GFP, a protein found from the bioluminescent jellyfish, Quaria victoria. Equoria victoria? That's pretty awesome. Anyway, bacteria that produce green fluorescent protein look green under white light and fluoresce under UV light. Assume that each research team has access to the following materials and is responsible for identifying one of the three plasmids. Note, you'll not actually use any materials during the pre-lab. Okay, before planning your experiment, answer the questions below to ensure that you understand some of the basic concepts involved in the lab. Some of these questions are specifically designed to help you think about setting up the experiment. Okay, so you can imagine a scenario on the AP exam where they're saying, oh, okay, you have this unidentified plasmid. How do you know what type of plasmid it is? So you set things up. So let's go through this. Number one, what is a plasmid? Pause. A plasmid is a small ring of DNA that has genes, usually something that helps with survival, again, like an antibiotic resistance gene. And it's important because plasmids can be shared. A plasmid is just a little extra ring of DNA that can be copied. When it's copied, then you can form those little conjugation tubes, and then bacteria can share the plasmids with one another. What is transformation? Well, again, transformation is the uptake of exogenous DNA. It's uptake of DNA from the surroundings. So with transformation, bacteria can actually move these plasmids from one cell to another, and it will transform the bacteria because it allows them to do new things. And the cool thing is that once transformed, those plasmids could be passed on to offspring, so then it becomes a change that is heritable. Okay? Number three, why is naturally occurring transformation beneficial to bacteria? Well, bacteria, they reproduce asexually, right? So it's really important for them to be able to basically create variation in order to survive long term. So being able to take up bacteria from the surroundings or from other bacteria and allow for transformation is really an important way to create variation. Why is transformation useful to research scientists? Well, it's a universal code, right? So if you can figure out how to use enzymes to sneak some therapeutic gene or gene of interest in to a bacterial plasmid, and you can move that plasmid into bacteria, then you can make bacteria produce the protein that you're interested in. So bacteria can be like a workhorse for researchers because it can make all sorts of stuff. All right, let's move to page S6. Number 5, should you plate some of your transformed bacteria onto plates with antibiotics? Why or why not? Well, here's the deal. When you go through bacterial transformation and you try to get bacteria to take up the plasmid, most of them aren't going to do it. As a matter of fact, very, very few bacteria will actually take up the plasmid. So what you want to do is be able to screen out any bacteria that didn't take up the plasmid. So if you have a plasmid for, again, say kanamycin resistance, any bacteria that take up the plasmid will produce proteins that sort of break down the kanamycin and allow the bacteria to survive. The bacteria that don't take that up wouldn't survive in the presence of kanamycin. So if you have a plate with kanamycin in the agar, it will kill off any bacteria that weren't transformed. Likewise, if you have a plasmid that has an ampicillin resistance gene and the GFP gene, what will happen is bacteria that don't take up the plasmid, if they are exposed to an agar plate with ampicillin, the bacteria will die. The only bacteria that survive will be the ones that take up the plasmid. And again, what will happen is they'll glow. The ones that take up the plasmid end up glowing. So again, the whole idea of growing bacteria on these plates with antibiotics is that it kills off any bacteria that don't take up the plasmid. That way you're only dealing with the bacteria that have the plasmid. Number 6, What would you expect to see if you plated some of your transformed bacteria onto a plate without the antibiotic? What would there be an advantage to doing this in terms of understanding the results? Okay. Well, if you don't put an antibiotic on the plate, then there's nothing that's going to select against the bacteria, right? It's nothing that's going to kill them. So you get what's called a lawn. With a lawn, you just get sort of like a bumpy film of bacteria across the top. So again, a lawn is just the agar will be covered with bacteria. And what that does is it serves as a control that simply shows that the bacteria, they survived your procedure. So when you do your setup, you're going to have some plates that just have luria broth, no antibiotics. And when you have the plates that just have luria broth and no antibiotics, ideally the bacteria all grow on that to show that they survived your transformation. They survived your experiment, because there will be opportunities to kill the bacteria if you're not careful. So again, it acts as a control group. Okay. Number 7, to transform bacteria with plasmids, technicians first make the bacteria competent or capable of taking up the DNA by placing them in calcium chloride and chilling them. Plasmid is then added to the competent bacteria, and the plasmid-bacteria combination is taken through a few more steps to make the bacteria take up the DNA. In your experiment, should you treat a tube of bacteria that you do not add plasmid to exactly as you do a tube of bacteria that you will transform? Why or why not? Again, this is creating a type of control group. So you'll have a tube with bacteria that you have a tube that has been transformed and a tube that hasn't been transformed, or I should say a tube that has the plasmid and a tube that doesn't have the plasmid. So you'll run sort of parallel experiments. You'll go through the transformation steps with a plus plasmid and a minus plasmid. So one group gets the plasmid, one group doesn't, and again, serves as a control group. Okay, now, oh boy, now we're on to the actual procedure. So beginning on page S7, what I would encourage you to do, like actually read through this, annotate, highlight, know what you're going to do. Okay, be familiar. Now, of course, it's a little challenging since you haven't seen a lot of this stuff. But again, there's going to be a little tube with the plasmid. So each group will get a plasmid. It's been assigned to your group by table number. Let's see. You're going to have some plates that are available. You're going to get those transformation tubes. Let's see. You're going to have ice available because you're going to have to keep some stuff chilled. You're going to get these inoculating loops. They're in yellow. Again, they're like the little wands that you'd use to blow bubbles. Transfer pipettes. So you'll have some pipettes here. And then you're going to get a vial of LB broth, which is some food, because after you transform their bacteria, you want to give them a nice meal. So we're giving some luria broth. And then there's that calcium chloride. Again, some stuff will be shared. I'll have a warm water bath in front of the room. We're going to share the incubator. We're going to share the plasmids. And then we're going to share those starter plates. So just sort of read through that. Okay, S8. This is where we jump in. One thing we need to do here is stress sterile technique. The whole idea of sterile technique is that you're covered in bacteria, surfaces all around the room are covered in bacteria, and only things we want to use are our special treated E. coli. Now, the E. coli we're going to be using have had genes for infection knocked out. So the bacteria have had any genes removed that would allow them to cause illness. Now, that said, we're using live organisms. Things can mutate and change. I've never, in I don't know how many years, 8, 9 years, I've never had anybody get sick, and I doubt anyone will. But you just got to be careful with stuff and use sterile technique. So the idea is everything is single use. If anything touches something it shouldn't, it's got to go. Okay. So first of all, when you're opening packaging, you always go to the handle end and you open at the handle end first. So for your pipette, you open at the bulb, you lift this out, and the tip of the pipette should never touch anything, okay? Never touch anything with the tip of the pipette. If you accidentally bump it on something, go straight in the trash, okay? You should never lay any of these pipettes down on the lab table, okay? You use the pipette. As soon as you use the pipette, go straight in the trash. We don't allow this to touch anything other than what you're required to use. Now, these culture tubes. The culture tubes, we want to keep them clean or open. Clean as you use stuff. So what I would, sorry, I tightened these up quite a bit to not let any air get in over the weekend. And boy, oh boy, is it tough to open up. So the way you'll handle these culture tubes, you hold the tube in one hand. With your other hand, you'll take your pinky and wrap your pinky around the cap and lift that up. Okay, so you keep the cap in your pinky, you keep the tube there. Do not let the tip of the pipette touch the sides of the culture tube. It just goes straight down, pull up what you need, straight back up. Okay. Again, if the tip of the pipette hits anything that it shouldn't be hitting, pitch it, get another one. Okay. So again, culture tube, wrap your pinky around the cap, lift up the cap. That way you don't set the cap down on the table. Okay. The cap never touches the table. You go straight in. Don't bang the sides of the mouth of the culture tube with your pipette. You just go straight down in, straight out, and then cap back on. That way nothing touches the table. Again, something touches the table, it goes. Same deal with the inoculating loop. When you go to use the inoculating loop, use the handle end, not the wand end. So you open it up just as much as you need to. Take that out. The wand here that you'll use to pick up your plasmid, this should not touch anything other than the plasmid, okay, or the fluids you're putting stuff into. If it touches the table, boom, in the trash it goes, okay? I know it feels like it's using a lot of materials, but it's more important to not, contaminate your samples. Okay, so let's sort of talk through the procedure. So you'll move through things. The first thing I want to do is encourage you to pay attention to volumes, okay? The volumes are really important in the lab. And so to help with sort of volumetric measurements, I've got like a little sample pipette here. We're using these pipettes. The instructions are in microliters, so it's millionths of a liter. 1 mil is 1,000 microliters. So there are gradations. I was looking at the pipettes. It looks like it says 0.5 mils. That's 500 microliters. There's a little dash below that. That's 250 microliters. That's a volume we'll use. And then there's like a little neck at the end of the pipette. That is 100 microliters. Okay. So I think you're going to mostly be using the 250 and 100. So the only thing they've marked on the pipettes are 1 mil and half a mil. So half a mil is the same as 500 microliters. The little notch on the pipette that you'll see after that is 250. And then 100 isn't marked. It's the neck of the pipette. And you'll notice where it gets narrowed there. So that's 100 microliters. So just make sure you're using the correct volumes when you're moving stuff around. All right, let's see. Looking at step 4, what you're going to do is basically pick up some bacteria from the starter plate. At the front of the room, I'm going to have these starter plates, and you're going to take this and use the starter plate. Now, here's the deal. When you have your starter plate, like this will be on the lab table, you do what's called clamshelling. When you clamshell, you only lift the lid off as much as you need to get in there. You don't pick up the lid and set it down somewhere else on the table. Okay, this lid should never touch the table. You'll have this plate, you lift up the lid and clamshell it, and then you take your clean loop and you go and whoop, 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 pick up some bacteria. Now, this is going to be grown on agar, which again is sort of like a jello. Do not poke the agar or scoop up some of the agar. Sometimes people feel like they have to grab a bunch of agar. You don't want to do that. If you do, it makes it more difficult for the bacteria to be transformed. It actually makes your experiment worse. So what you do is you just place the loop on the surface of the agar where the bacteria are. The bacteria looks sort of slimy and yellow, and you just glide it along the surface very gently and pick up a little bit of bacteria. Now, how much do you need? It says about half the size of a pencil eraser. So if we imagine this is like a pencil eraser size, you only need about that much. What I've seen people do in the past, we've got bacteria growing all over a plate. They gather huge amounts of bacteria, and then there's not enough for other groups. Okay, we got to be thoughtful about our use of bacteria. So you'll scoop up just about a pencil eraser's worth. Okay, you'll see like a little clump of bacteria. You don't have to like fill this entire loop with bacteria. Oh my gosh, that'd be way too much. On the agar, it's a space about the size of a pencil eraser. You can even go 2 if it make you feel more comfortable. a couple pencil erasers, scoop up that bacteria. You're going to see a little clump of bacteria on the end of that loop. That's all you need. Okay. So you scoop up that bacteria, and then you'll have the culture tube with your calcium chloride in it. Sorry, again, I tightened it down. You may want to loosen these a little bit before you try to open this. There we go. So you'll remove that. And then you're going to have your bacteria and then this chilled calcium chloride. So what you want to do is you put this down in the culture tube and then you spin as fast as you can. Okay. So you spin this, and when you spin this down in the fluid, it'll knock the bacteria off the loop and into the calcium chloride. After you've spun this should look a little cloudy. Okay. So the calcium chloride initially is going to be clear. It's going to look like water. You're going to take the bacteria, spin that down in there, and it'll look a little cloudy. The bacteria should be dissolved off the end of the loop as well. Okay. Number 5. Now, as quickly as you can, what you need to do, the bacteria will sort of be in clumps when they come off the wand here, and you don't want clumps of bacteria. Again, you want these to be separated. So you'll take a clean pipette and you'll suck the bacteria up whoop, into this, and then you push it out, and that resuspends them. So you work really quickly. You suck up the fluid, push it out, suck up the fluid, push it out. You do that a few times, and then again, it should be nice and cloudy when it's done. When it's all cloudy, you're good to go. Eventually, then, you're going to get a new loop, and you're going to add some plasmid. So the plasmid is in a little vial. They're going to be numbered. What you'll do is you'll get your little vial. And here's one thing I recommend. The vials will be really little. Tap the bottom of the vial on the table, because sometimes the plasmid is up in the cap. So students will be like, the plasmid's all gone. Well, as things get handled, the plasmid sometimes winds up inside the cap. So the easiest way to do this is take the little vial and tap it on the ground. Take the cap off. Again, don't set the cap on the table. Take the cap off, and then you're going to place this clean wand down inside the vial with the plasmid. You pull it out, and it should look like film on the end of a wand when you're going to blow bubbles. Okay. If you don't see a film here, you didn't get the plasmid. So tap the vial with the plasmid, take the cap off, down in you go, pull that up. If you have film, you got your plasmid. Okay. Then the plasmid will end up going in the culture tube. Now when you get to step 10, after you've added the plasmid, you got to give the bacteria some time to take it up. So you're going to have 15 minutes of chill time. So that's sort of nice. Yeah, it's working quickly. Then you're going to wait for 15 minutes. All right, so now let's move on to page S9. This is step 12. It says, following the 15-minute incubation on ice, we have to heat shock the bacteria. So we need to heat them up. Now, I'm only going to have it at like 42 Celsius. We don't want to kill the bacteria. So I'm going to heat them up a little bit. So for the heat shock, timing needs to be very precise. It's at exactly 90 seconds, okay? So you bring them up on ice, like they're going to be on ice in a cup. You're going to bring them up, transfer them to the water, 90 seconds, immediately back on ice, okay? That's one step that's really important, because if you heat the bacteria up too much and don't cool them back down, you'll kill them. So you'll have your cup of ice, bring them up, move directly from the cup of ice to the heat shock, and after 90 seconds, immediately back in the ice to cool them back down. All right, now after you've heat shocked your bacteria to get them to take up the plasmid, you're going to give them a nice meal. So in step 13, you're going to give them 250 mils of luria broth. So you'll feed them. And then it says to give them 5 to 15 minutes for recovery. We found that 5 minutes works just fine. Plus, with our length of our class periods, we got to go with 5 minutes. So in step 13, 5 minutes is great. During the incubation, you're going to add some glass beads. You only need 3 to 4, okay? Now, one thing I recommend when you add the glass beads, here's the thing. The auger on these plates, it's sort of springy. It's like gelatin. So if you add glass beads to the auger, it'll bounce out, and then you got beads all over the place. So what we found works really well is to have this upside down. If you turn your plate upside down and add your 3 or 4 beads to the lid, close it up, flip it right back up, and then you're good to go. Let's see. Now, after you've done your work here, when you get to step 14, groups, or group number one, you've actually got the glow plasmid. So in step 14, You're going to spread the bacteria with the plasmid around. You're going to make your plate that'll have glowing bacteria. Everybody wants their bacteria to glow, right? So I'm going to have some extra plates that are LB amp plates. They have the ampicillin. And if you're in one of the groups that has plasmid number 1, what I ask that you do is just go to your neighbors and take some extra. You're going to have a little bit of extra fluid in your plus plasmid tube. Take a little bit of the fluid from your plus plasmid tube, share it with a neighbor. And so if you're using plasmid 1 and you have your plus plasmid tube, you're going to have the glow gene in there. If your neighbors also get access to a little bit of that plus plasmid fluid in an LB amp plate, they too will be able to have the glowing bacteria. So again, just make sure we're sharing, and that way everybody gets to have some glowing bacteria at the end of the day. All right, step 15. This is going to be at the end of class on Monday or Tuesday. We need to store your plates. So what you're going to do is take all of your plates, you're going to stack them together, and you're going to take some tape and wrap some tape around the outside. Now, what's important is that you store these upside down. You want the lid at the bottom because you can sometimes get some condensation on here. And if you have this turned upright and the condensation drips down on bacteria, it'll kill the bacteria. So we're going to store these things upside down. So we'll incubate them overnight. You guys will come back in on Wednesday or Thursday and see what's growing. So I have the information that we discussed. I've sort of written out, procedural reminders, plus just some general stuff. With the general stuff, it's things like, please make sure you wipe down, because of COVID, wipe down the equipment before and after you use stuff. So anything that'll be reused, please wipe it down. Make sure to wash your hands when you're done with the lab. Be very careful about volumes, especially the 250 and particularly the 100 micromils, or microliters, I should say. Make sure our lab stations are neat and organized so that people can use stuff. And then again, if you're plus plasmid, you've got the glow gene. So when you get to step, I think it's 14, not 4, share your plus plasmid with a neighbor who has an extra LB amp plate. And again, I've got notes here on all the stuff. I'll have that up on the board. So be prepared, because as soon as your group is here, I want you guys to get started, because you're going to need every minute of class. And divvy up the work. This is something where you can't have one person doing everything and everybody else is sort of checked out. You all need to pay attention. You all need to double check one another, and you all need to be in communication with one another. I've only had a couple of groups in all my years not get this stuff to work. And the reason that people are successful is because they're working together, they're communicating with one another, and nobody's taking the day off. Everybody's sort of helping ensure that everyone else is on board and everyone knows what's going on. If you have questions, let me know, but come in prepared and ready to go, and you should be all set. If you have any questions, shoot me an email. Otherwise, I'll see you soon, and we'll transform some bacteria.